Alright, let's go into our laboratory for Unit 10, titled Genetics. This is a pretty straightforward group. I had to rewrite this. That's why it took me a little bit longer to get this. I'm doing the transition, and I'm hoping that it'll make the experience a little bit more enjoyable. You can find any of the materials for this particular lab in the laboratory as you go to it on Blackboard, and you'll see the listing. Right now, the only one that we're really doing right now is the genetics. So the materials are there and it's ready. We have the teams. One of the ideas that if you wanted to collaborate on a section of this lab is writing a report or a PowerPoint presentation on one of several types of diseases. I think there's like 7 for you to choose from, and this gives you a collaborative tool that you guys can work together on. It's optional. You don't have to do it that way, but it might be a challenge. It might be fun to do. So Unit 10, the genetics. This is what we're going to look at. We're going to talk about genes, genetics, loci, alleles, and the various things. We'll be getting into Mendelian genetics in class part next week. And so the lab is actually going to provide sort of an introduction. I like the duality. It helps reinforce everything on both sides. And so we're going to go over the Punnett squares, talk about sex-linked inheritance. We're going to talk about blood types. That's a biggie. Everyone should know about blood typing and what the restrictions are, and what happens if you mix blood types. And so those are some of the things that we're going to talk about. And so let's get into it. So we already talked about in class that genetics, the genes, the heredity that make us up, come from our parents, of course, 1/2 from our mother and 1/2 from our father. And some of the terms, vocabulary is genes, heredity, the locus or loci. That's where a particular gene resides on the chromosome. We have 23 pair of chromosomes, and we have alleles. Now we have one copy from our mother, one from our father. So of a particular gene, we have an allele. We have one form of that gene, let's say for eye color, and then we have another gene for eye color, and then whatever is dominant will be what is expressed. And so alleles, it just means of the same gene. Alleles are from that gene that may, let's say for eye color, you may have blue and green, and it depends on which is dominant that's expressed, or you may have a mixture of the two. It depends, but those serve as an allele for that particular trait. Some alleles are, as I mentioned just moments ago, a dominant or recessive. If it's a dominant type of trait, and then that means that would be expressed. The recessive traits means that they may be stepped on, or not being expressed, I guess is the best way to put that. Alleles are represented by letters, and the convention is the dominant is always the capital and recessive is always lowercase. So if you're looking at what makes up the genetics as a representation or symbols, well, that's the genotype. When you're talking about the genes and the various aspects of that, you can have a heterozygous trait or a homozygous trait. So we have homozygous, heterozygous. Homozygous means that the traits are the same, or the characteristics are the same, so the two alleles are the same. And heterozygous, it means the two alleles are different. So this is different. and homozygous is same. So you may have big B, or little b. Those would be homozygous for a trait. Big B, little b would be heterozygous. And then it depends on the dominance of that trait. So the letters representing this person's genotype would be homozygous for the dominant trait. This would be heterozygous for that's a dominant trait, big B, little b, so it's a heterozygous. And then little b is homozygous for the trait that's not being expressed, or the recessive trait. So this should be review, if not first time. We're going to go over this again in class. The phenotype is what we actually see. This is the physical manifestation of whatever the genotype is encoding for. You can see it starts to get really sort of technical as we go into this, but little b, big B, little b, big B, all would have some sort of phenotype associated with it. These two perhaps would be expressed as the same. And that happens a lot. Even though the genotypes are different, the phenotype would be the same. Little b would be expressing a homozygous recessive trait, and phenotypically that would express as something different. Because of the dominant gene, excuse me, it's been a long week. I think it has been for you as well. I hope everything's going well for you. So, a survey of genetic traits is what I'd like you to do, and of course you can do this together on Teams. It's not mandatory. I'd just like you to get familiar with Teams, and I will post a copy of this on Teams. If you wanted to use this and just chat using the chat feature in Teams amongst whoever you want to share the traits that you might see. So if you look and read through the instructions, they're written here, and you look at the traits, and you just, do you have attached or unattached? A lot of this can be done easily over the video, and the descriptions of the traits are there. Ultimately comes down on this page here on table 1 that you record the sample size. Is it just you, or did you get with someone else? Do you show the dominant or recessive trait? And all of that's written up in the text preceding. And then you can just say it's dominant or recessive trait, and you can use the lettering that they provided. Your personal phenotype, usually you don't know if it's recessive or not. Like it would be big T, little t. If you have tongue rolling, it very well could be big T, you just don't know. But if you have the recessive trait, then little t, and that's what you would use. Oops, that goes in that column. Of course I would make that mistake. Personal phenotype is that you can roll tongue, let's say. I can do that, and so that would be big T, or big T, not sure which one is which, and I'll erase those there. And so rolling tongue, and then that, and then you just follow that down and do those for the traits a little bit more on the next page. And it helps you get used to using the nomenclature and representing recessive and dominant traits. So all you need to do is to fill in that table and save that table. I've given a Word version of this document so you can enter that information pretty easily, and then you can send that to me by email, and you can do it all together if you prefer, with all the sections that we have assigned here. And so the very first part is to do the personality type of testing and your traits. And next is the blood typing, and this is an important aspect, and I've written up a little bit more detail than the original manual because it is such an important aspect. And I'll go over some of this, but more detail is available to you if you are interested in that. So the type of characteristics that we see with blood typing is a codominant. In other words, you have two alleles, both will be expressed. Where in standard dominance type of thing, only one or the other. In blood, we've noticed that it's a codominance, and they express both of the alleles. So if you have the allele for the various blood types, there's only three really that we A, B, B, and then the fourth type is one that doesn't really express anything. It's called type O. These are the types. I say there's really only three that are associated with a particular antigen that's expressed. If you don't have an antigen expressed at all, it's type O. So a total of 4 of the different blood groups. So the interesting concept of this is we have the genetics that shows if you have an allele with A and another allele with A, you'll be type A. Or if you have an allele with type A, and if it has the allele with nothing superscript to it, that would be assumed as type O. So type O is I. B would be IB. A would be IA, and then would be I, A, and B. And so those would be represented like that. And the idea is that this would be homozygous for the O trait, this would be heterozygous, but it would be for the B trait and then the O trait. AB is the heterozygous for A and B, and they're both expressed. So you can kind of follow what that is. And this is important because if you were, let's say, to get in an accident or something and you needed blood, it's good to know your blood type, and they have to match that blood type, because if you get the wrong blood type, then bad things happen. We have rejection against your own blood, and matters don't get very much better from there on. It could be a life-threatening situation if it's not dealt with. So all of the things that I just discussed are written up here. Here's the 4 different blood groups, and this is the phenotype of the blood, and these are the genotypes of the blood. And of course we also do it for forensics. If somebody leaves a blood type behind, a blood splatter or something like that, we can type it, and it lets us know what's going on. Also fraternity type of issues can be done. So type O, you can look at type O. Now I don't know if you know your blood type or not. Normally in the lab I would test it for you. It would just be one sample to see if it matches what your parents might be. But while you are at home, or if you can find out from your parents what their blood type is, your mother and father, you'll be somewhat of some expression from the two, and it'd be interesting to figure the genetics out for that. But if you already know, that's great. If you don't, and then the next time you go for a doctor's visit or donate blood at the American Red Cross, might be a good way to find out what your blood type is and then help someone else at the same time. They are in dire need of blood right now, and so if you can do that, it'd be great, if you're not sick, of course. So type O lacks both the A and B antigen, like I mentioned. It's the only one that is really, there's the 3 different types that have the antigen, the A, the B, and the A and the B. If you are devoid of both of those antigens, then you're type O. And there's some interesting aspects of type O, and there's a lot of things that have been talked about with that blood type. It's the universal donor, so because it doesn't have any of the antigens that causes the immune response, type O blood is really preferred in terms of donation, because they can use that blood with any person that needs the blood. The other aspect I'll go ahead and mention is something called the Rh factor. And the Rh factor is really from genetics a long time ago with Rhesus monkey. There's some cross-reactivity, and the Rh factor is a minor type of protein set that's on the outer of the surfaces of red blood cells. Normally, it's the major group is the A antigen or the B antigens being expressed on the surface, but the Rh factor is a minor protein that's expressed, and it also becomes a factor. So it's usually plus or minus, or Rh positive or Rh negative. It also can be referred to as type D. This is the more current nomenclature for Rh factors. Your type D. Are you type D positive, type D negative? So I'll just go ahead and mention that. Type O negative blood, that's the safest one. It just, for whatever reason, no one really knows. For the coronavirus right now, if you're type O, it seems to not to be as deadly or as infectious for type O. Of course, I'm A positive, and that's the one that's the most deadly for, I guess. Lucky me. Anyhow, you can the type O blood is the universal donor, and it's quite interesting to see what blood type you might be. Type A blood has the A antigens, and obviously the type A blood is the antigens being expressed and making it's the most common, the second most common blood type in the country. So the type A, I guess I have one of the more common, the second most common. Patients with this blood can donate to individuals who have either A positive or AB positive, so we can give to the AB positive folks. O positive or O negative we can't give to, because obviously the O does not have the A, and that could be dangerous for that person. Type B blood, B positive forms of blood are rare in the United States, and it just seems to be the way it is. 9% of Americans have B positive, while only 2% have B negative. So you might have some suggestion as to what your blood type. It might be O or A or AB. I've done a fair amount of testing in the lab with students, and I see A and AB the most common in the lab with my students at Lake Tech. So those with B negative blood can donate patients with AB or B positive. See, if you don't have that particular antigen for the Rh or type D, then if you're negative, then you can give to either. But if you're positive, you can only give blood, which doesn't matter what it is, A, B, or whatever. It has to be the positive recipient. So type AB, of course, express both of the alleles. Blood classified as AB contains both the A and B markers, or the antigens on the surface of the cell, and as a result of that, we don't have antibodies against them. Now, just really quick here, what an antibody is, it's a protein, sort of looks like this. They're bound together. The antigen binding region can be found at the ends of the Y fork at one end of the antibody. So this is an antibody, and they call this region here the FC, or the constant region. The only reason I mention that is that we have antibodies that can stick to that particular antigen of whatever, an antigen, and as a result of that, the immune system tags it, and then all sorts of cascade events will occur. And if we have blood of a different type that's given to us, we'll actually create an immune response which includes these antibodies, and they will stick all over the blood that we're not supposed to have, and that flags the immune system that, hey, we need to get rid of this. Well, we don't want to be getting rid of or attacking blood, but unfortunately, if there was a transfusion error, the nurses have to stay attending to any blood transfusion, and if you see purpura or little dots of black and blue forming under the skin, that's a Arthur's reaction against the blood, and they stop the transfusion and infuse Ringer's lactate or some water of some kind that's adjusted for right osmolarity and all that, and try to dilute it out. But it's a very serious event, so doctors take it very seriously. So the rarest type is AB in America, although I've seen, oddly enough, in class. AB positive blood can donate there to any of those that have AB positive. It's one that we have is the universal recipient, because type AB can take blood from anywhere, A or B or O and all that. The only thing we have to worry about, of course, is the type D factor. So this is the universal recipient. So if you were to have a car accident and you're type AB, then you're pretty much good to go. You can find blood either type. So the RH factor is written up. I've already discussed that, and you can look through it. I threw in, when I talk about these antigens on the surface of the red blood cells, I just kind of throw that out there. So I included a picture of what they actually look like and what they're made of. I'm not going to ask you, but I just figured that you might like to know. It is a single gene that represents, of course, as we've already mentioned, we have different alleles, A, B, and if it doesn't express this antigen at all, type O. And you can see the information as a curiosity. And if you're interested in looking at 3D images and the like, I provided an example where you can go and explore for yourself. And in the written documentation, and you obviously don't have to copy this and write it out, you just select it when you get to your document and put it in your browser, and you'll get a listing of various variations of the blood group antigens, and you can click on any one of those. In particular, click on where it says 3D view. Free browser, any Chrome works really well, but pretty much any browser will work. And then boom, it brings you into an environment that's 3D. You can take your mouse and rotate this around and look at various things. And if you look at the various settings, set style, if you set it to, there's a setting, I don't know if you can see it there, maybe not, it's called space filled. And space filled will give you the image that looks like that. And it's really cool that you can change the parameters. The only other parameter I usually change is, instead of just having a rainbow of colors or kind of random, I tell it to select the subunits, and so we have color. So you can see this is made up of 2 subunits. It's the same drawing as you just looked at, except now it was colorized just by a click of a mouse. To indicate where the 2 subunits are, and there is the active region right there that binds and all sorts of things. So anyhow, this would work for any protein. It's all free. I just thought it was a good introduction for you just to kind of play around. It's free, it's available to you, and it's on the PDB databank. If you just do a search on PDB or hit the link that I just gave you on the previous page, and then you can enjoy looking at any type of protein. You can type in, there's molecule of the month in the PDB databank. If you do that, then they look at some various proteins that you probably will recognize the names of, and you can do the same sorts of things with it. And explore. It's a great way to learn more and go more in depth about some of the proteins and structures that we talk about in class. Okay, so the action item, I guess, on the blood group is I had a chart somewhere in here. I think it's just to read the section. I'd be happy with that, just to kind of read through it, and you know your blood types, because on the lab practical we'll be asking for that. I thought there was a chart for you to fill in, but there's not. That's okay. So as far as the blood grouping, if you have questions on that, feel free to ask me about it when you get a chance later on. So shut this off here. Okay. So if we'll move on from the blood typing. Now we'll go into a section called Mendelian inheritance, and I alluded to this at the very beginning. Gregor Mendel was a monk. This is really strange. He was trained as a classical as a scientist, and because of financial worries and things, he joined a monastery. And in those days, the monks would itinerate. In other words, they would go from one monastery to another on a regular basis. And this clever man who was curious about doing something in addition to just what a monk does, whatever that is, of the day, he would go from monastery to monastery. He said, well, gee, you know, what he ended up doing was experiments in plant hybridizations. And it was really interesting. Now, you have to understand, at this time, and a moment in time in 1866, they didn't have a clue about genetics. They didn't know anything about that. And so his idea, one of the things he brought up was this concept of traits. All living things have traits or characteristics. And he used his skills, and one of the skills that he obtained was math. Through his training and research, he looked at things from a mathematical perspective in terms of percentages and that sort of thing. And that was really what he sort of brings to the table. If you're interested, I'm a history buff. I love looking into the history of things. More importantly, I like reading about the scientists and their backgrounds and who they are as a person. I tend to remember things better that way. That's why I always ask my students, you know, what they want to do and who they are, because those things interest me, and we want to find where, you know, my job is to get you where you need to go, at least a little bit that I can do. And anyhow, I posted the paper. One of the original papers was translated, of course, from Mendel's work, but I love reading through those things because it gives you an insight as to how he did things and what he did and why he did them. And so if you're interested, it's not, I'm not assigning it or anything. And that's the thing, is part of the job I guess I have is to try to find things that spark you or interest you. You may not be interested in biology, and that's fine, but it is about life, the study of life, and maybe there are certain things that we talk about that at least piques your interest about something. And I just want you to feel free to know how to go and look into things. You just never know. My daughter wasn't interested in biology and a career in biology, at least that's what she told me all the way through high school. But then she went in to NC State and did the exact degree program that I did, and which really shocked me because she hadn't talked about it. And not only that, she did it magna cum laude. And wow, really? She did it better than I did and got a full scholarship to go to Emory. And it's those sorts of things that I hope, if that's what you want, can happen for you. Is that Louis Pasteur is a famous microbiologist, and he always had a quote saying, "Chance favors the prepared mind." And the idea is you prepare your mind to see and observe things that might interest you and become your career. This is the paper of 1865 that I posted for you if you want to look through it. History is just one of those things that when it's written down, this was, I think, one of the important events in our knowledge of what's going on. So what he did was that he studied various plants, and let me go back to this page here. You could see this chart, and he very luckily, I guess you could say, or chance favors the prepared mind. He chose traits that were very standard and stable and followed a simple type of genetics flow. Had he chosen something that had some weird genetics like incomplete dominance, or I will talk about some of the others in class, but he didn't. He was lucky and picked very clean traits, and you can see the differences, and he was just tracing those. And that's where the concept of traits, he assigned some sort of letter to, as the terms of what the letter represented, like maybe tall and short. So T would be tall and little t would be short, that sort of thing. And this is the other sort of, nobody really talks about it, but he did provide that for us. So the gene, of course, is the basic of unity of heredity, and what Gregor Mendel taught us was that he came up with two basic principles: the law of segregation and the law of independent assortment. So the genes segregate for a particular trait. In other words, you pass them along. He didn't know what they were called genes. I guess I shouldn't say that, because at the time he did not know that. He just said it was a trait. And the law of independent assortment means that it random. It was totally random, you would see. But there was just only a certain number, a limit of traits for a particular thing, like I was showing you here with the plants having the color that they did, or these particular traits, that those would be logged and tracked and followed. And what he found was they would have the law of segregation, that these always seem to segregate together for this particular trait. And which was, nobody did this stuff in those days, and this was helpful for farmers when they were tracking traits of maybe a good milk producer over another cow that wasn't as good, and then they try to figure out, then what we do is make these cows that are good milk producers with others. Those concepts, some of the basic ideas, came from Gregor Mendel, and it helped that industry quite a lot. So this idea of the law of independent assortment is actually comes and translates out to something called the Punnett square. And the Punnett square is an important tool that we use to find out how things get transmitted. And so it's a kind of steppy. We're going to go over this in class, but and nomenclature, you have to be able to see the difference of capitals and little. So you notice my handwriting is absolutely superior to most handwritings, which is not the truth at all. But what we want to do is to try to write clearly when we do these types of tables. I found when I took standardized exams and things that if my handwriting was sloppy, I didn't do very well with these type crosses. So I slowed down and make sure. So if you notice, like the capital T and the lower T, I crossed it, made it look like that. So I just try to do things that favor as to what the dominant or recessive traits look like in terms of the characters that I use. So the other is that you always try to define what the letters represent, and I write that off to the side so that I can always remember that. So then you have to find out the parent. And the parent one could be the father and the mother. Is usually, it doesn't matter. It doesn't matter as long as you denote as to what they are. So if one parent was big P, little p, and of course whoever wrote this has told us that's the case, and the other parent, the female, was big P, little p, then you want to know what the potential offspring. Now again, this sort of is important if you wanted to kind of tell what your offspring might be later on with traits that you know about in your family. And so the same kind of genetics follow. This is what Gregor Mendel did in the 1800s. It's just amazing. Although he didn't understand it was genetics. Big P, big P, big P, little p, big P, little p, and then little p, little p. And so what I try to do is to accent the fact that this one's uppercase and these are lowercase. So all of these three here, phenotypically, what we see would be the same. because the dominant trait would be the dominant expression, and we see that. But the little p, this one here would stand out. It would be different phenotypically, and it would have the recessive trait. So typically we see 3 to 1 type of ratio. This is what his math showed him. He didn't understand why, what was going on, but that was the first of all, the law of segregation, that these genes will arrange like this, although he didn't know they were genes, and then the law of independent assortment. So these are the possibilities of the offspring, and that they would occur 3 to 1. So there's 3 times more likely that you'd have certain characteristic over 1. And that's the Punnett square. And so you can see it's nicely written out below here after I scribbled all over the handout. Does that make sense? I hope so. So to test that, or to give you the idea, instead of having an actual ear of corn, I could mail it to you, I guess, but why would I want to do that? Why would you want it? So I provided an extra copy somewhere in your package here. There's a larger picture of the corn, and all you need to do is to count down, let's say 3 or 4 rows that you can meet, and that's what we would have done in class or in lab anyhow, and just count the number of black ones versus yellow, and based on that, and just fill out the chart and come up with, if you want, big B is black and little b is yellow, if you want to do that. It's just as a suggestion. You can use whatever characters you want, and so assign those, and then you can find out what the possible parents might have been that provided the corn. Do the best you can on this. We're going to go over this again in class, but it's a great introduction to get you going into it. So this is called a monohybrid, just one cross. We just take one trait and cross it. There's another example with the Wisconsin fast-growing plants, a little bit more detailed. And what you do is you take a petri plate and have a circular piece of Wattman blotting paper. You wet it and put the seeds on it, and then after a couple of weeks they start to grow. So magically, there they are. I provided a picture for you, and you can see light green and dark green, or purple actually is the color. And then you can actually look at the stems, because the stem colors are purple or green, and you can write your data. Now, the table I fixed in your copy will be on the next page all together. But what this means is just fill in the actual the counts as you go through for each of the stems and seed lines, and I'll conclude that, and then you can come up with a cross. Now I just have you work through these analysis type questions best you can. See if you can figure that out. Next week I will go over this again with the results, and so that we can match to see if the results that you got were the same. Now, the very last part of the to-do list for this this week, which obviously I give you a week to complete this, but if you're real busy or whatever, I'm going to be real flexible. It's about when you turn this in, and you can work together on it if you'd like. You can call or use Teams. I would recommend using Teams on this one if you want. I'm just asking you to make a PowerPoint presentation and just choose one of these type of scenarios. It could be cystic fibrosis, Huntington's disease, sickle cell, Downs, celiac disease, and just do a PowerPoint on one of these diseases. Now you can work by yourself, or you can work in groups of 2, 3, however many you want, and you go online with Teams. You just say, hey, look, you know, you put a note in the Teams area and say, hey, look, you know, I would like someone to go in with a project. So if you're monitoring Teams, let's say, just pick the standard lab time that we normally meet for lab, and that way everyone can go on Teams at that time, and then perhaps you can pick someone. this is the first time. I don't like particularly teaching online type of things in the lab. I just, I find it a challenge, but that's okay. My nature is that I don't complain about it. I just meet the challenge head on and go on with it. And anyhow, this gives you an opportunity. It's amazing some of the reports that I've written in school. I still remember to this day. I did one on, unfortunately, on the avian coronavirus of all things, and how it transmitted and all that. So, and I still remember that report, and look how relevant it is today. I knew quite in depth about how coronaviruses are. They're a very strange virus, and they have certain, there's feline coronavirus, which is cats and various things. And so it helped. And these sorts of things will stick with you, and it's good to know. If you know something that might be in your family, it's good to do a report, or if you don't know anything about it, it might be a good choice to do, just to do that. So you write that up and just send me a PowerPoint, or you can post it on Teams if you want. I really like to email. That way I kind of guarantee that I got it from you and there's no question about it. And then the final is the work through the unit review questions, like we always do at the end, and work through those best you can, and then send that in. So you can just send the whole package, because you get to keep the original, and that way I can give you credit for it. And go through these, because these of course will find its way on a lab practical, and I'll go over it at the beginning of the next session, so you have that. So that is so the deliverables for this group is really quite straightforward. As you go through in teams and things, and however you guys want to work together, you can do that. Looking at the traits, fill out the chart on the back here, and then read through various the traits to figure out what if you're dominant recessive for that trait, and all that information's in there, and fill that out. And then the blood typing is more of kind of just going through and reading what the blood types are. It's important to be familiar with those. And then you can go through and go online if that interests you to look at the 3D stuff, as the antigens for the blood groups are there represented as proteins. Then the Gregor-Mendel inheritance, if you go through and read what we had just covered, and then what we're going to do is to work across with the corn as a monohybrid cross, and then you get your data here, and then try to figure out, the idea is a 3-to-1 type ratio. The Wisconsin plants, you have the data here. Just fill out the data and count it up, and then see if you can work out the genetics on that, and I'll go over that next week. And then the case studies. Now this I'll give you until the end of the semester to complete, to get the case studies done. And all it is just a PowerPoint, however you want to present. We'll leave it up to you as exactly how you want to do it. You can even do it as a info chart if you want, instead of a PowerPoint. But the idea is to convey what just choose one disease, and you can work in by yourself or in groups of 2 or 3, and do that, and then fill out the review questions. And that's it for this lab. I hope it's not too painful. I had to restructure it so it kind of fits the format that we're working on here. But again, I'll make sure you wash your hands off and limit going out, because we have that ban on there, and the idea is that we don't want to spread the water droplets of this thing, so we stay at home and hopefully ride through all of this. I know it's it's quite strange, it's an unusual circumstance, but we'll get through it. And just be assured that I'll be here to go over and talk about this. If you have questions, shoot me an email. I have my office hours. You can use Zoom for that. I like Zoom because it's easy to use and allows a little bit better handling of multiple people, so you don't you just show up during the office hour if you have some questions, or you just want to say hi or complain or whatever. Get some of the boredom at being at the house off your chest. Anyhow, stay well, and I'll talk to you next time. And please take care.